Okay, you guys, this video is going to go through meiosis, and meiosis is the formation of sex cells, which are known as gametes. And what happens is you start off with a diploid number, one cell, and you end up with 4 genetically different haploid cells. So here I'm showing interphase. So there is one, and remember the DNA would be more like chromatin. It would look like this. All right, so that's how it would look. We're really going to focus on the meiosis 1 and meiosis 2. All right, but just to review, there's a G1, okay, where the cell grows. There's an S phase, that's where the DNA duplicates. And so what I would want you to do while you're following this video is I would want you to do the same thing with your pipe cleaners that I gave you. So what you're going to do is you're going to go ahead and duplicate. And they are considered sister chromatids now. Remember, this is the centromere. The waist is the centromere. And so this is what they would look for, and they go into the G2 phase. All right. You have too long and too short. Right, and 2 different colors. So you have a long, for me, a purple, and a long green or magenta. And this would be paternal, meaning from the father, maternal from the mother. Paternal, maternal. All right, and so we are going to go into prophase 1. So I'm going to move these out of the way. And I have a lot of things written down here. See if I can put them together for you, but you should be following along in your study guide. And this starts on page 103 and 104. And so the homologous pairs, so the pairing is called a synapsis, and they come together like this. Okay, and these are homologous pairs, meaning they have the same size, they have the same shape, but most importantly, they carry the same genes. So I called that, you can see I have these labeled in different spots. I have these are homologous chromosomes. That is the same as being a tetrad, right? When they're 4 together, and the parental is homologous to the maternal. And, so if it's eye color or height that this chromosome is carrying, okay, so is the other one. They're homologous to one another. The pairing of how I showed you that they are coming together and pairing up is called the synapsis. And remember, the waist is the centromeres. All right, the other thing that happens is crossing over. And this is a source of genetic variation. So I have a little arrow right here. So this is the chiasma where they cross. So I have my finger on where the cross is. So they cross over and they exchange parts of their chromosomes. And this is why you're genetically different. So you're going to take some of your loose pieces and you're going to demonstrate that they have exchanged pieces. And it happens on the lower part too. We're just not going to model that. This is a little bit easier if we had the other chromosomes in class, but I don't have enough for everybody. So we're going to do it to your short ones as well. If you want, you can use your beads. That was my idea, but because it's faster than this, but I don't think we have enough of all the colors of the beads to really show it accurately. All right, so there's a lot that goes on in prophase 1. So the crossing over is a source of genetic variation. So, if you need to pause the video and go through and figure out all the things that happen in prophase 1, go ahead and do that. When you are ready, we're going to go ahead to metaphase 1. You'll notice I didn't talk about things you should already know, like the chromosomes are now visible. They're condensed in prophase 1. The spindle forms, the nucleolus disappears, and the nuclear membrane disappears. Right, so those things still happen. I'm really just going over the main things that happen in meiosis. So we're going to go on to metaphase. I labeled my paper metaphase 1 and anaphase 1. Spindle is completely formed, and your homologous pairs, or tetrads, are going to line up at the equator of the cell. They're the middle of the cell. So that should be a little bit easier to see. All right, so something else that happens in metaphase 1, and I'm just going to write this over here, is independent assortment. And what this means is that this set of chromosomes is independent to this set. All right, so what these tetrads do is different or independent than what these do, how they line up. So parental, maternal, parental, maternal. These could also line up as maternal, parental. And so we have 23 pairs in our chromosomes. So each pair would be independent and can line up in any format. And that is going to give us more genetic variation as well. After metaphase 1 is anaphase 1. And in that anaphase 1, they're going to come apart. And this is known as disjunction. So I'm going to put disjunction over here with my anaphase. I wrote that on the paper over here. So the separation of when they come apart is disjunction. It looks like I lost some of my chromosome here, so I'm just going to fix it real quick. And something that can happen, I will tell you, is something called non-disjunction. So we're going to get to where we talk about this. I'm just going to leave these like this. So this I'm going to model with these small ones. So they should come apart and become sister chromatids again. But what can happen is they don't separate correctly for whatever reason, and you can get this. So I have 1, 2, 3 over here, and I only have 1 here. So that can be a problem if this ends up being an egg or a sperm. It's going to have less chromosomes. Or over here, if this is the egg or sperm cell, this is going to have too many chromosomes. Right, so that's known as non-disjunction. Next, we are going to have, take yours out, next you're going to have telophase 1 in cytokinesis. So you are going to have your sheet of paper, and we're just going to put them like this. They are sister chromatids again. They are considered to be haploid because we consider them diploid as when they double, and they were 4, and now they're 2 each. It's a little confusing. So cytokinesis, your nuclear envelope, nucleolus reappear. The pinching in happens. The division of the cytoplasm, cytokinesis, also happens. All right, so this is a reduction. Meiosis is often known as a reduction and then a division. So now we're going to go through the whole thing again with 2 cells, and it's basically the repeating mitosis, basically, is what's happening. So we're going to get now, you're going to draw 2 cells. You want to keep track of yours. So here's mine. And I know mine aren't totally inside the nucleus, but they are still sister chromatids. They were 4, now they're 2 each. Okay, so this is prophase 2. Same thing happens as spindle forms, nucleolus disappears, nucleolus disappears. That's prophase 2. That's it. Now we're going to go to metaphase 2 and anaphase 2. So you're going to line up at the metaphase plate, the equator of the cell, and that's going to be your metaphase 2. Okay, line up at the middle. Anaphase 2, they're going to come apart, and that is known as disjunction. All right, and what can happen is non-disjunction. So this could happen, and this could happen, and there could be 2. You couldn't see that. So this could happen where you get 2, where these 2 don't separate, but these 2 do correctly. So this cell here would be extra chromosomes. This would be less. All right, so showing that again, it should be this, but it could be like this. And that's called aneuploidy if you get an abnormal number of chromosomes. Okay, so that is anaphase, or metaphase 2 and anaphase 2. So I'm going to keep these separate. And then I'm going to go to telophase 2 in cytokinesis. So what you're going to have is you're going to have 2 chromosomes in each part here. And I think you're getting the idea of what's happening. You're going to have the pinching in, telophase 2. You're going to have the reformation of the nuclear envelope and the nucleolus, the pinching in, the cleavage furrow of the cell membrane. The division of the cytoplasm is cytokinesis. And then when you are done, you're going to have 4 genetically different haploid cells. So this is like crazy drawing that I did the other day real fast, but you're going to get the idea, and that's what's important. So you're going to draw really fast you're going to draw 4 cells that are genetically different. And I wrote the N equals 2 because there are 1, 2 chromosomes in each daughter cell. So you have 1, 2, 3, 4 genetically different haploid cells. We started off with 4 chromosomes. Each daughter cell has half the number of chromosomes. Now, we'll talk about this, but remember, these are the formation of sex cells. So one of these could turn into the sperm, and that could be what fertilizes the egg. So fertilization, you're going to get 1 egg, and then you're going to get a sperm that come together. So that's another reason that there's genetic variation as well. All right, so what you'll want to do is make sure that you set yours up and you're able to go through this and you are ready to go for next class and you understand how meiosis works. Remember, you're getting 4 genetically different haploid represented by N cells. 4 genetically different haploid cells. All right, and that is it.